Hello, everybody. Welcome to the MDC Connects webinar series. I'm Sarah Brockbank. I'm a lead scientist at Medicine's Discovery Catapult, and I'm hosting the session today with my colleague Dan Bond. We're now on to the fourth in this series of weekly webinars that run every Wednesday at 2 o'clock. That's it. As I say, today we're in the fourth. This is a series of 9 informative drug discovery sessions, and each of the sessions is delivered by experts at the Medicines Discovery Catapult and our CRO network partners. Each of the sessions is focusing on an aspect of preclinical drug discovery, and we started with selection of the right target and identifying the hit, and today we're optimising the compound, before thinking about PK/PD, disease models and efficacy, and finally defining safety and biomarker strategies. So in this, that's the fourth session, we're thinking about optimising the compound, and I'd like to welcome 3 speakers. So firstly, Al Dossetter is the co-founder of MedChemica, and Al is going to talk about in silico drug design, with some pointers for what to do and, importantly, what not to do. Then we've got Richard Weaver, who's the CEO of XenoGesis, and he's going to focus on the ADME properties of the molecule, and again is going to highlight key issues and common mistakes. And finally, Juliana Maynard, who is lead scientist of the preclinical imaging unit here at Medicine's Discovery Catapult. Juliana is going to demonstrate how imaging technologies can be used to help us understand the PK and biodistribution of our compound. So we will take questions at the end of each of these presentations. If you wish to ask a question, can you type it into the Q&A box at the bottom of your screens, not the chat box, and the speakers will address them at the end of their talk. So over to you, Al.
Thank you very much, Sarah. Just clicking and sharing my screen on, and I will just start my presentation into presentation mode. Thank you very much for giving the opportunity to talk here today. What a challenge on talking about optimising the compound in a short talk. So I'm going to focus on what to do and what not to do, and this is driven kind of out of experience. In this presentation, there's a lot of literature references, and if nothing else, I tell you today, if you go and to follow us on Bucket List Papers or go to our website on Bucket List, these are the 100 papers you have to read, not during the pandemic, but sometimes in your early career or during your career in kind of drug discovery. So we think. Agree with us, disagree with us, say they're good, say they're bad. But anyway, many of the references I have in this talk are in that bucket list. We generally add about 2 a week, so we're at 50 something or other at the moment. This pandemic has given us actually a rare opportunity in that there's a community project actually searching for SARS-CoV-2 inhibitors at the moment. Inhibitors are one of the proteases. So please follow us at COVID Moonshot. There is data to look at, and you can see how the project's going live. Very, very rare moment coming to. So just a couple of Twitter feeds there for you. Sarah's introduced me. Thank you very much. And what I'd like to do today is I'm a medicinal chemist by choice. I'm a trained chemist. And I did 13 years in a large pharma and then started MedChemica in 2012. And we are an in silico drug design AI company. We spend about 30% of our time doing consultancy work. We are supporting COVID Moonshot at the moment, but we also support antibacterial projects as well and oncology projects. But given the nature and the length of this talk, I do want to focus on just some line of lessons I've learned. I can't do justice, and I could talk for an entire day about in silico drug designs, but I'm going to outline what the problem statement is and why we do in silico drug design. And take you through this agenda of some of the cheminformatics work, stuff you should always do up the front, and give you an idea and a feeling and a lessons learned from understanding when we do predictions where these kind of come from in 2D design. And I've really got one lesson from, maybe 2 lessons from 3D design, to take you through. And principally, I'm going to illustrate this with a project example, because there's nothing better than looking at a project example from back in the day called cathepsin K. I do have a second project example, 11 beta HSD. So basically, if you'd like the longer presentation and more details, give us a shout and we can do something. So why do we do in silico drug design? I'll let you read those statements at the top. It's a packed slide. We went through a period in the noughties where we made lots and lots of compounds, and there was a notion that if we made lots and lots of compounds, we'd get to the compound quicker and it would be like a sausage factory. We'd get more candidate drugs out at the end. What we've realized is we now need to be more sensible. In silico techniques are there to help us to analyze the data we have well and unbiased, and refine our compound design so we do fewer iterations to get to a better candidate drug and in fewer iterations. So let's crack on. That's what we're trying to do, that problem statement. So I'm assuming you're familiar with design, make, test, analyze, and the next speakers will talk about test, and they'll also talk about the quality of data. Where we are in silico design is analyzing the output from these tests, bringing the information together, helping idea generation, helping rank the ideas in the design phase so we make as few compounds as possible. And not only as fewer compounds as possible, but going around that design loop in fewer iterations. Here's another whole load of references. This slide shows being recorded, and there'll be slide sharing at the end of this. This is all about optimizing and encouraging the clarity in that process. Where we are now in 2020, as chemists, we get bombarded with an enormous amount of data. And this is where computers come in. They help us process all of this data. And very importantly, it's not just a volume and velocity problem. It's also being unbiased as well, because we have to acknowledge that we're human beings as well. Hopefully you've read some of those areas that are an influx of data that leads to structure-activity relationships in our molecule and analysis, that we've got to then think of the ideas and then rank the ideas of where they come from in amongst the pressures of time and competition and the rest of it. So in terms of what to do and what not to do, I've already alluded to one not to do, and that's to be very, very aware of rushing at the analysis process. Be very aware of yourself and cognitive biases that we have as human beings, and actually let the computer help you with that analysis. And one of the ways, and this is incredible, this is a very old technique, 1964, Free and Wilson published one of the first mathematical models to help automate structure activity decisions. And essentially all this is, and I've cut and pasted this straight out of the paper, is working out what each group on the molecule, say like this phenyl ring over here, makes a contribution to in terms of the potency of compound. And the simple idea, if you add up all those components, the sum of them, that predicts your IC50 or your LD50 or your EC50. I can tell you, over the years, as I've gone on, there is nothing more reliable than the Free Wilson analysis. And I'll show you an example in CAT-K of how we did this, and just made a few molecules and made an enormous progress on the project very, very quickly. We have a slightly more sophisticated, more automated method of doing this now, and we call it permutative MMPA. And one of the things you can do with this, that we can do with computers, is to bring data together from multiple sources, from patents and the literature, look at the relationships between molecules in the computer and remove the bias element for it. Here's an example with two patterns of kinase inhibitors. The top sort of blue colored compounds are the ones from one patents, and the bottom ones are another set of compounds. And that's how they're interlinked together and how they cluster together into very similar compounds we can analyze. If you take this and you work in this way and you combine Free Wilson and these types of analysis in an unbiased way, and here's another project example, we can take this set of data and analyze which are the key groups, if you like, the warheads on the molecules that not only are good for potency, but also give you as what we call more bang for your buck. And there's another learning, not just unbiased, techniques that stand out as these two warheads did here, to say these chemical changes give you a large increase in potency, but also less of a penalty of things like lipophilicity as well. So in silico design, number one, very good for analysis, lots of analysis, pull in lots of data, be unbiased, be rigorous and more quick. Wow, that's just one of several techniques. There are a lot of techniques in 2D and 3D compound design now to actually come up with new things. I have noticed that in, if you want to know about the likes in 3D of force field simulations and free energy perturbations and quantum mechanics, I'll put some names there of people you could ask to do that. I'm sure I know Cressett's talking in another one of these sessions later on. I'm going to hopefully mention conformation and torsional analysis and the great resources and databases there are, which means you don't have to reinvent the wheel. Good news, every time you go and use calculated lipophilicity, you are doing in silico drug design. Calculated lipophilicity has been around for quite a while. It's a way of estimating the lipophilicity of a molecule. We have for a long while, and this is a challenge that always comes back to its silico drug design, that, oh, but it's all just lipophilicity. No, not true in my case, not true in my experience. But I did want to leave you with that thing of it's important lipophilicity, and every time you use C-log-P in selecting your designs, you're doing in silico drug design. I'd like to bring you to this reference here from Mike Waring. We've known for years there's been a sweet spot in the middle of lipophilicity, either log D or log P, kind of between 1 and 3. And what really we've learned is if you're too polar, you get one set of problems. If you're too lipophilic, you get a set of problems, okay? That doesn't mean that reducing lipophilicity will solve everything for you, and it isn't in my experience, because many times I've been between 1 and 3 in a compound, I've had a suboptimal compound. There's a great quote here from Pete Kenny, where describing how it's not just a panacea for fixing everything. And what I've learned over the years is that structural modifications and keeping in mind size and lipophilicity of what's really important in compounds. But we fall into this trap in analysis quite often because of the programs that we have. And when we fill a plot of data up with our data, and here's some examples out of the literature. The left ones, honestly, is one of mine. I've fallen into this trap. The program likes to fill the square with your dots, and you often draw a line, a linear fit, if you like, linear regression fit onto this, if you like. And the angle of it's 45 degrees, which makes us think we have a strong relationship to lipophilicity. But if you actually look at these, and particularly I bring your attention to 3A4 in the middle, the slope of this line is only 0.16. So to get a significant change in 3A4 inhibition, you need to drop a lot of units of lipophilicity to achieve this, which is impractical, very difficult to do it, and you'll get another set of problems. So always plot on a linear scale, don't bin your data, and make sure you spread your units out to see the true reflection of the slope of that line. And this is often a thing where people do analysis. This is honestly one of my own. I've never published this one. And people bin data and say, I'll set cutoffs of molecular weight and lipophilicity and I'll count the number of compounds. Don't do it. I haven't got time to describe it in future, but essentially what this slide says is a set of data up to one time point gave one picture, and a set of data doing the same analysis gave a completely different picture. And what it illustrates is just how dangerous it is to do binning. And you can make the computer tell you that it's statistically significant if you want to. I'll bring you to a paper called Inflation of Correlation. So like, there's another lesson learned there. But what I want you to do is appreciate and how QSAR models are built. And I'll bring you to your attention to this paper on the method of calculating a C-log-P. This one's based on atoms, and it's the same principle. You count the number of atoms of a certain type that make a contribution and add up all those contributions. And it turns out A-log-P 98 is quite good. Well, one thing I need to implore on you is to go understand kind of how a model's built. They're all kind of like this one way or another. Going forward, and remember, they're only predictions. They all have errors involved in them. So that is one of your problems of using cutoffs. Don't use cutoffs because you're at the fortune of the error. going forward. But appreciation of any in silico design is an appreciation of the errors in any of the models we do. And the next two speakers, again, will talk about the quality of data leads to the quality of models, which is what these two references are. I haven't got time to tell you about max molecular pair analysis. So skip straight over and try and bring that together very quickly in just a couple of minutes with a project example of cathepsin K. This was a structure-based design project. We had fantastic crystals, so it was perfect for 3D design. And we had a pretty awesome start point straight out of screening, which was a fairly active compound. Selectivity could have been improved. We were already a high molecular weight, and we had clearance problems. So a kind of a good few things to fix, but nonetheless, it was quite good. We did an incredibly rare thing. That top compound there, 095, we stripped everything back to the smallest possible compound we could make. And it was 300 nanometers, so we lost a whole load of potency. And then as a result of having that compound, we could do the free Wilson analysis that told us about very, very simple R groups. And now we could look at these and then pick out, actually, that one's quite good. That one gives us more potency than you'd expect for the lipophilicity. And the crystal structure started to make sense. Okay, so this was our first step back, make simple compounds. In my experience, and still my ongoing experience as consultants, the simple compounds are never made. What does this go? Why have we got all these groups over here? Do we need all this bit? What does just phenyl do? Okay, so simple compounds improve your silico models, simple compounds improve your understanding. As I said, there's a structure-based design program. I've got 2 minutes left, here we go. I only want to tell you one thing about 3D molecular design. When you get a crystal structure back, ensure you sit down with a crystallographer that solved the crystal structure and look at the electron density of that structure. And that's what this wire mesh diagram is here. The amino acid groups and the structure of your potential inhibitor is fitted into that electron density. They are often far from perfect. And you can see on this left-hand one, this steroid is just blobbed in there into this blob. If you look at leucine 402, you can see a bit of the leucine just sticking out. There isn't any electron density to go with it. Once you start looking at these, you realize just how difficult this is to get a quality crystal structure. On the example on the left with two phenol groups on either end, how do we know which way round that group is? I've seen a lot of people and a lot of projects waste a lot of time because they took on faith the crystal structure was correct. And if you look at this electron density, you can make a very good judgment about it. Again, fantastic references that go kind of with this one. We have brilliant crystal structures that went with this one, and we were pretty confident and we could predict very well how these went into the cathepsin hole going forward. That slide there, which that's there really for you to share with you and look at later on when you've got the slide decks or watching the video back. Read some of the references about applications and limitations and the kind of learning, and two great references on optimizing intermolecular hydrogen bonds and things. But in short, we did a matrix of 55 compounds, again, based on this free Wilson analysis. I'll just go through that, to flick the builds on very, very quickly. And we quickly obtained two good series of compounds after this. And this is finishing off. We ended up with this carboline. It beautifully wrapped round in that crystal structures. We knew we only needed to add substituents on one side of the group. There was no point making any others. They wouldn't make any difference. And on the top group, it allowed us to modulate the physical properties and the metabolism. And that's how we ended up with the methoxy group. The other series we optimized, and just very quickly. This dimethoxy group was a breakthrough. A lot of people tell you this is a bad idea or get metabolized and etc. etc. If you go looking for this in crystal structures, it is not the shape you would predict from the simple Vesta model we're taught as organic chemists. It's actually this flat shape with the methyl groups pointing out. And the only lesson I want to tell you is there is a wealth of structures in the protein data bank and in the crystal databases, which means you don't have to go and measure these things or get a new stricture, you can get a shape in it. Once you've got that, you can design compounds that are the same shape and move forward. So there we go, very, very quick example. I'll skip over these and just go straight to the conclusion slide to wrap this up. Oops, went too far. So we're there in silico drug design to do fewer iterations, get as much as information you can. Free Wilson analysis is one of the best things you can do going along. Make sure you plot things on a continuous scale. Understand how models are built and their limitations. Get the best data you can to build the best data models you can and make simpler compounds. PDB, CCDB, great treasure trove of information. And don't forget to check that electron density and check your root mean square errors before you do any drug design. Thank you very much for listening.
Thanks, Al. Let's take a couple of questions.
Yeah, where's the Q&A box gone? Whoops, my Q&A box just disappeared. Can somebody read the questions out? That for some reason my Mac has put the QA thing half up my screen and I can't actually read them very well.
Sure. So I've got a question.
Yeah.
Angelo Silva, with current AI technology, what is the estimated percentage of molecules that can escape in silico analysis?
Have you got an hour? Sorry, don't foresee one answer. Yeah, so the analysis of error is very important. There are certain functional, I'm going to give a Donald Trump answer. There are certain groups we do very well at predicting at. I'll tell you what they are. They're small changes around a benzene ring. If I had time, I can show you a plot with the errors on lots and lots of functional groups that we at least have a measurement for, and show you and say that, and I'd put it at like well more than half at least. We can't do accurate predictions on. I would predict, and I've written about this on LinkedIn, the company that wins in AI is the company that invests in data. And it is possible to look at the data set you have and do the analysis that goes, where are we short of information? What functional groups would we like to know about? And then go out and get that data. And it's not going to take 1 million data points. It's not going to take 100,000. It might take 10, 20, 30,000 new measurements. And you could be the AI company that's the best AI company in the world.
Okay, Al, I think we'll move on to Richard now. Thank you.
Good afternoon and good morning, everybody. So yes, I'm going to now talk about some DMPK. Just a quick note to say that we have had some internet issues from here, so if I drop out, hopefully I will be back. Okay, so first of all, just a quick introduction to XenoGesis. So we are a CRO based here in the UK at BioCity Nottingham, and we offer in vitro and in vivo ADME, bioanalysis and pharmacology. And we also do quite a lot of consultancy and modelling as well. Our clients are far-reaching around the world. And again, we're a team of 33 people here, and I'll leave you to read through that at your leisure. So what I'm going to run through today is something which is very important, intrinsic clearance, and something called IVIVE, in vitro in vivo extrapolation, and also when it doesn't work. I'm going to also talk about data quality, and Al really touched on that, and it's not always the case. And actually, when data is good, it's amazing how things can suddenly make sense. I'm going to talk about plasma protein binding and why there's still some myths that are being believed there, and then finally finish up with some formulation slides and how we can increase bioavailability. So, intrinsic clearance. What's the point of optimizing it? So see, some of these are statements that I've heard over the years and also repeatedly hear these. So for example, we have compounds with high in vitro clearance, but low in vivo clearance. I have compounds with high oral bioavailability despite high clint. We have spent 6 months optimizing our compounds to have low clint, but actually we go in vivo and the clearance is very high. There isn't a correlation between in vitro clearance and in vivo clearance. And also, we use microsomes because hepatocytes are too expensive. And then also, I've read literature that says hepatocytes are poor. and I'm measuring in vivo PK anyway, why should I bother? And then another one we get often is, well, it's just cheaper to do the PK in the Far East. So here's an example. Here's a client. They've plotted intrinsic clearance against observed clearance, and perhaps surprisingly, there's no correlation. And they might say, well, they don't predict, you know. And then, so what are the options? Well, option 2 is always science, and that's what we would always recommend, and start to look into this data for them. So just a quick, brief recap on what these things are. So intrinsic clearance is, I say, it's an in vitro measurement, and it's the volume of incubation of media from which all drug is removed in a unit time. And it's usually corrected by the number of cells. Most importantly, it's a closed system. There's no flow, and the only binding you have in there is to the cells or to the plasticware. In vivo clearance is different because that's the volume of plasma or blood, usually blood, as that filters through the eliminating organs. It's removed per unit time, and we correct that for body weight. Most importantly, it's the unbound drug that is eliminated, and that's important. And plasma protein binding obviously forms part of that equation. So actually, if we apply something called the Wellster model, there are multiple models here, but if we apply that and correct everything for unbound, so unbound intrinsic clearance, correct for plasma protein binding, suddenly the correlation's starting to look good. But on this plot, there are 2 obvious outliers. So we significantly underpredict these 2 compounds, and I guess the question is, why is that? So then we start to look at the science. And obviously, the standard hepatocyte assay, that measures loss of compound due to metabolism. But that isn't the only route of clearance from the body and elimination from the body. So for basic, neutral, permeable compounds and that sort of sweet spot of log D, log P that Al talked about, then we tend to get that. But for acids, polar compounds, low permeability compounds, often hepatic uptake and renal clearance will dominate. And it's important to mention that the total clearance happening in vivo is actually a sum of all those different parameters. Now, of course, we can't actually measure hepatic uptake in the standard assay. We can't do that because we sample both cells and media, and therefore drug that is taken up into the cells isn't lost. However, if we do 2 experiments where we do a regular assay, and then we also do an experiment where we measure loss from the media alone, then we can start to see some differences. And a classical example here is the statins, and this is some data that we generated in our lab with atorvastatin, where you can see in this traditional assay in blue, then actually we're just getting a, sort of what looks like a first order process, elimination. But actually when we look at media, you can see we have an initial faster rate, followed by the lines then paralleling. And that initial rate is actually the compound going into the cells very, very rapidly. So then you might say, a client might say, well, I've used the Wellster model, I've corrected for this renal clearance and uptake, but actually I still get a poor correlation. Now I just want to talk about data quality. So we often review third-party DMPK data and PhysChem. Sometimes the data is fine, very often it's fine actually, but sometimes it's absolutely not fine. And the first thing is the intrinsic clearance. That's the one experiment that we review that time and time again is wrong, I would say, in our view. So we've optimized our assay, and actually you can start to see the data on the right. So we've taken some literature compounds where clearance is liver and hepatic, and on the top diagram we've plotted some directly from the literature. And actually when we use our assay, then you can see the bottom line and things start to make a bit more sense. So that's the in vitro side. There's also the in vivo side. Is the PK correct? And it's amazing how many times the PK data is not correct. And one example can be from iron suppression, and I can talk about that offline or take a question on that. Another example is enterohepatic recirculation. Again, to the uninitiated, or if it's not blindingly obvious from the profile, this can lead to an apparent lower clearance and higher volume of distribution. So here's a real life example. A client came to us and said, actually, we've got a massive in vitro disconnect here. The intrinsic clearance in hepatocytes is very low, but it's huge clearance. So this is the left-hand side. We started to look at that and said, yeah, absolutely, it looks like a big discrepancy. But actually, we were expecting metabolism for these compounds, hepatic metabolism. So we were surprised that it didn't add up. So we recommended repeating some of this data. We repeated the intrinsic clearance for the client, and we also repeated the in vivo clearance because there were some flags from that data. And actually what you can see in the table in the middle, this is now the predicted clearance by the third-party data, predicting to be 22, but their observed was 142. When we used our data, we predicted 40, and actually when we did it, it was 46. So again, actually, the predicted clearance is absolutely correct when you use the right data. If we'd have believed that data at face value, we may have gone off on a wild goose chase, for example, looking at hepatic uptake or other routes of metabolism, renal clearance, for example. So I think it's just important to say how important data quality is. Right, quick one on plasma protein binding now. So free drug hypothesis, I should say rule, that's what Dennis Smith says, and he's right. In the absence of active transport, the free drug concentration in non-eliminating tissues is the same at steady state. And we often hear these things, I've heard these in all my career. We need to optimize PPB. Increasing PPB will give us a better PK. Increasing PPB will increase my half-life. Lower PPB is greater free drug exposure. That's true, but it's really important to note that plasma protein binding does not affect free drug exposure. And that's the most important thing. We should optimize free drug exposure and not the free fraction. So again, people might then say, okay, well, if that's the case, why on earth should I bother measuring protein binding? Well, you absolutely should, because you need that to translate in vitro to in vivo data for a start, to do that IVE. And very importantly, you need it to compare across species. And this is about toxicology now. So it's extremely important to understand if you have a higher free fraction in human, for example, free fraction versus the animal species. For example, if it's 10 times more free in human versus the tox, you need to know about it. You've lost the margin there, a safety margin. We measure the plasma protein binding in the same animals that we conduct the PK. As differences can be very significant, and that's really important. So again, just to be very clear, just on this, I could talk all day about this, but it's very important to know what the plasma protein binding is, but it shouldn't be and can't be optimized. So let's talk about bioavailability now. So this is a 3D plot. It might look complicated, but on the one axis, the vertical axis here, you've got increasing bioavailability. On the x-axis, you have increasing absorption from 0 to 1. And then on the z-axis, you have an increasing fraction escaping hepatic clearance. And again, I could talk about this in a lot more detail, but just by increasing the absorption to 1, i.e. 100%, it's still possible to have 0 bioavailability, as you will see on the left. And what's really important is actually understanding what does escape hepatic clearance, and also obviously the metabolism in the gut as well. So start there before you try and do any formulation. So just as an example, a real life example, a client came to us and they wanted a formulation to enhance exposure for use in tox studies. We reviewed this data. It had very poor aqueous solubility, as many compounds do these days, unfortunately. It had, reasonable permeability. That was pretty good. But there was also, not only that, there was a plateau of exposure as well at up to 20 mg per kg. The clearance, actually, when we reviewed it, was very low, so very low, less than 5% of liver blood flow. And doing the maths, it's easy to do this on it as a paper exercise. If all the compound is absorbed, then the bioavailability should be greater than 95%, should be possible. And actually, what we realized in this particular example, that actually it's the absorption bit that's limiting the bioavailability to do with solubility. So what did we do? We reviewed this, as I mentioned. We had a look at anticipated dose in humans and did some gap analysis. And with, at the time, it was with a company called Juniper Pharmaceuticals, which is now Catalent. And we did a parallel screening strategy. So we did lots of different formulations. We chose 2 in the end, the 2 circled there. And we screened these in rat. And we did it very quickly and with not much compound. So for the whole process, we only needed 5 grams of compound for all these different formulations. we're able to turn everything around between 8 and 12 weeks. And this is the data. It was a nice roaring success. So the graph on the bottom with the blue, remember this is on a log scale, so that's really important. The standard bioavailability was terrible at 2%, less than 2%, but actually the two formulations we looked at were fantastic and greater than 80%. And very importantly, it was dose linear. So that's important to note as well. So not only do we increase the bioavailability, but we also had great dose linearity from 50 to 500 mg per kg. And of course, that was important because it allowed this project to progress. Otherwise, it would be dead as a dodo. You could not do tox. So we rescued this for the client. Just as another example of trying to showcase something, we chose raloxifene again to actually showcase spray-dried dispersion technology. This is a compound that does still have some clearance from the liver and also the GI, but nonetheless, there's something you can do about that to increase its solubility. And again, we actually looked at some non-spray dried formulation. This was in rats, and also some spray dried form, and then the spray dried formulation. And you can see we actually increased the bioavailability by nearly 7-fold with a very, very simple study that we could understand completely from a DMPK and also from a PhysChem perspective. So again, very nice success, and we presented that at various conferences. So a bit of a flying visit today. I've probably got a couple of minutes left just to give you a bit of a taster. But actually, you know, this is my view, the team's view on how DMPK people should influence drug discovery. You know, I'll stand on my soapbox and say that, you know, DMPK is not a service provider in drug discovery. It's an integrated process, and we should influence across the way. So we should identify the key DMPK issues. I'm a chemist also. Look at the structures first of all, show me the structure, and then we'll have a look and we can say a lot from that. We should be able to help design better compounds, fix bioavailability issues, that's true as well, and hopefully extend release to rescue poor half-life compounds. That's possible as well, extended release as well as increasing bioavailability. You should always predict PK and doses early. I've not talked about that today, but that's really important. And it has multiple knock-on effects. For example, if the compound is a 10 milligram predicted human dose versus 1,000 milligrams, that's very different with regard to API scale-up, demand, cost of goods, etc. It's important to prepare your asset for due diligence, assuming that you are a biotech here, and most drugs, this is where they come from these days, and you'll be probably either going it alone or selling to large pharma. So it's important to do that, and also assess the DDI risk. And the DMPK influence should go all the way along, you know, all the way along to phase 2A, at least, I would say as well. So at that point, I'd actually like to say thank you very much, and I'm happy to answer a few questions.
Hi, Richard. I just want to take a question or two.
Sure. So I've had a question here: how strong PEG can suppress iron during HPLC analysis? Significantly. I say it's not HPLC itself, it's actually the mass spec side, obviously. Significantly, I published on that way back in 2002, I believe. So, you know, clearance can change by 5-fold, for example. So a huge effect. And then I have another question about PPB, a great question. What happens if your compounds are greater than 99.9% bound? This is a real issue, a real challenge, and obviously it's a challenge to measure that accurately. There are ways to help and do that. I know AstraZeneca are about to publish on that, and obviously you can do dilution, you can do other techniques as well. So if anything, to be able to have compounds that are less than that, so you can measure them, that is a good thing. And maybe I'll stop there if that's correct, Sarah.
Yeah, thanks, Richard. Okay, Juliana.
Fantastic. Thanks, everyone. Okay, I'm just going to share my screen. Okay, so hopefully everyone can see that. So thanks for giving me the opportunity to talk to you all today. So my name is Juliana Maynard, and I'm lead scientist here at the Medicines Discovery Catapult. So today I'm going to follow on from the excellent presentations that we've already heard and discuss some of the technologies that we're using to show and demonstrate biodistribution and pharmacokinetics of a candidate compound. So here in MDC, we're a government organisation funded by Innovate UK. We've been tasked to accelerate drug discovery. So we want to get drugs to market quicker, and those drugs that won't get to market fail quicker. We want to build that link between the target and its biological effect in a translatable system that will reduce preclinical attrition of drug discovery. So we want to ensure that we have the best models, the best biomarkers, and the patient selection strategy. So within our discovery science and technology department, we have a series of focus areas to do this, including complex cell models, target engagement, biomarkers, and our focus for today in preclinical imaging. So using imaging in drug discovery. So our overarching value proposition, as we see it, is to provide a visualization of biological functions to accelerate disease understanding and drug development. And there are a wealth of different imaging modalities available, and all of them offer unique and various solutions to answer the biological questions that we're trying to answer. So we can categorize these into different subsets. We can look at organ accumulation and distribution. So if you inject the drug into the body, understanding exactly where it goes to. We can look at target expression and engagement. So does the drug hit and engage with its target as well as just getting there? What's the optimal biological dose? So pathophysiologically, are we actually altering the biological state? Toxicities, is the drug safe to be used? And do our preclinical studies predict what you see in the clinic? So today, my focus on this webinar is on one of these subsets and how we can use imaging to assess organ accumulation and distribution of a candidate compound. So why is assessing organ accumulation and distribution important? Well, quite simply, it enables us to track exactly where a compound is travelling in an experimental animal or human. And it's an important assessment that is commonly from small molecule drugs as well as complex medicines. And the challenge we have is in quantitatively determining that percentage of administered dose in specific organs throughout the body and at specific time points. And as I say, that's important for both small molecules, and with small molecules, this can be done in other ways as well. But for larger molecules and complex medicines, fully quantitative methods just aren't available. So the solution, we believe then, is that in vivo and ex vivo preclinical imaging offers a compelling approach and a solution to non-invasively assessing that compound distribution and accumulation, moving compounds towards clinical translation. So here at the Medicines Discovery Catapult, we have imaging expertise in multiparametric imaging modalities, and these range from functional radiological imaging techniques such as positron emission tomography, so attaching a radio-labeled moiety, computed tomography to assess anatomical information, high-frequency ultrasound, bioluminescence near-infrared imaging, so attaching a fluorescence or a bioluminescence ligand to visualize distribution, and also ex vivo imaging techniques such as mass spectrometry, DESI, and MALDI. We have our brand new digital spatial profiler and our advanced microscopy imaging suite. So offering solutions across all areas from functional to anatomical assessment, and complementing this with our molecular pathology capability. Underpinning this, we have our home office and radiological license, our facilities within Alderley Park, and we're completely disease agnostic. So we work across naive, oncology, fibrosis, thrombosis models, and we really try and combine all of these together to be multimodal, so we're allowing an enhanced interpretation of functional and molecular data. So today I'm going to go through some of the examples and case studies of how we can use imaging to understand that distribution of a therapeutic. So as we've said, the rate and degree of drug distribution is complicated because it depends on many factors. It depends on blood flow, capillary permeability, and protein binding. And an administered substance must go through a series of cascades in order for it to be effective. And maximal efficiency of each of these steps is very important to optimize that therapeutic benefit. So when the compound is administered, it will circulate, it will accumulate, it will penetrate, it will become internalized, and it will be released. And tracking the process of that from the systemic level to the cellular level becomes essential in understanding and characterizing that therapeutic. And imaging offers many ways in being able to do this. So the first example I've got today is using fluorescent near-infrared imaging device to perform longitudinal Psi-7 biodistribution imaging. So near-infrared imaging itself holds enormous potential for in vivo imaging to assess that distribution of a candidate compound and characterization of the biology. It allows the ability to have deep biological penetration within the body and also being able to image that whole body distribution with good signal to noise. So nowadays there are more and more dyes available, and by labeling with that near-infrared dye makes it possible to track and study the distribution of the molecule. So for an example, the image here we have on the right-hand side is looking at an antibody and being able to longitudinally assess that tumour distribution. So looking at what the penetration and accumulation of the antibody is across time, but also where else does it go within the body. And you can see that longitudinal assessment of images in the images here. So sometimes you may also want to understand not only that your compound gets to your disease area, but also does it get to anywhere it's not supposed to. So this is an example using zirconium PET to assess off-target toxicity. And off-target toxicity can often cause issues with promising therapeutics and being able to progress these further. So zirconium PET is a very good tracer because of its half-life. It means you can inject it into the body and then repeatedly image several days after injection to be able to longitudinally assess that distribution of the compound. So here we were able to show in our case study that we had no non-specific binding and that a promising antibody-drug conjugate could then be attached and have a cytotoxic payload added to it without causing any unwanted toxic side effects to any other areas within the body, only the area that it was targeting within the tumour. In a very similar way to the Psi-7 near-infrared labelling, where we're measuring full body distribution, we can also use zirconium PET to assess with high sensitivity and specificity tumour penetration longitudinally. And by performing PET imaging with also assessment of biodistribution, so using our gamma counter, we can assess that distribution and concentration down to a picomolar level, so get very, very sensitive changes and an understanding of the accumulation within the tumour across time to be able to see what's happening throughout the body. In the nanoparticle and complex medicine space, it also offers huge potential. So two major obstacles that are facing cancer nanomedicine are the tendency of nanoparticles to be taken up by normal tissues because of their size, but also of their ability to efficiently penetrate the tumour itself. So here we have an example in the case study of our collaboration with the University of Leeds and their therapeutic microbubble and targeted ultrasound platform. And what we wanted to show as a catapult was the superiority of platform to show that it was better than liposomal encapsulated drug alone, but also understand the mechanism of action of the platform itself. So what we were able to see was that there was indeed enhanced delivery of the microbubbles to the tumour as a drug carrier, but also that accumulation and delivery was predominantly through the reticular endothelial system, as we had expected from this nanoparticle formulation. And these are the kind of studies and understanding that drug delivery and distribution that meet a key demand of regulatory guidance within the nanoparticle space. Another very important area which has an unmet need is understanding blood-brain barrier penetration. So the blood-brain barrier prevents the brain uptake of most pharmaceuticals. So certain small molecules can cross via a mechanism known as lipid-mediated free diffusion, but these properties are lacking in the majority of small molecule and all large molecule drugs. But there are several ways to test whether your compound has crossed the blood-brain barrier, and we see that the imaging again offers opportunity in this space, not only to be able to non-invasively image blood-brain barrier penetration, but also, as I say, to be able to do this longitudinally and get that dynamic time course of distribution. So I've just chosen 3 promising ways in which we can do this. So we've spoken through examples of how we can use near-infrared and PET to assess whole body distribution. But in the blood-brain barrier space, we can also look at PET and near-infrared here. On the left-hand side image, we have an image of using PET to assess antibody distribution and penetration in the brain. An exciting area for PET imaging with the blood-brain barrier penetration also is the use of dynamic PET, and complex modelling also offers a huge opportunity of really being able to quantify and assess that dynamic penetration within the brain. The near-infrared imaging and bioluminescence probes also offer a very non-invasive method, as well as mass spectrometry imaging, and that offers huge potential to define drug distribution in very small brain structures with a non-labelled approach. So moving on then to mass spectrometry imaging and the potential it has in the drug distribution space. So there are two main mass spectrometry imaging techniques: desorption electron spray ionisation, known as DESI, and matrix-assisted later desorption ionisation, known as MOLDI. So DESI allows chemical information to be obtained from a wide range of surfaces. And here, mass spectra are collected by, as you can see on the image on the right-hand side, by rastering a spray across the surface and then computationally assessing that mass spectra that you've gained. MOLDI is the use of matrix-assisted desorption ionisation, in which samples, which is often a thin slice of tissue, is moved in 2 dimensions and a spectra being recorded as it moves across the actual tissue section itself. So here I've just chosen an example of how we've used both DESI and MOLDI to measure and assess tissue drug concentrations. So the image on the left-hand side here is a DESI image with an H&E stain, and this shows drug concentration on the surface of the skin. With the image on the right-hand side, and a higher resolution MOLDI imaging that confirms what they've seen with DESI, and you can also see that drug distribution on the surface of the skin. So a very non-invasive method, it's very label-free. You can take the tissue ex vivo. As I say, you can also combine what you're seeing in vivo with the ex vivo to really build that bigger picture of what you're seeing within the tissue itself. So my final case study that I wanted to go through today was the evaluation of the blood-brain barrier penetration of a polymeric nanoparticle through confocal microscopy. So this is using our steroid blood-brain barrier model. And with this model, we've used this with drugs that are known to cross the blood-brain barrier, so idarubicin, and those that are not known to cross the blood-brain barrier, doxorubicin. And what we showed here using our confocal microscopy techniques was that treating with that polymeric nanoparticle, we could really visualize and understand the dynamic time course of blood-brain barrier penetration of this nanoparticle itself, and really characterize this for the client that we were working with. So just to summarise then, it's a very whistle-stop tour of the capabilities that we have and where we've been using this within the drug distribution space. But we really believe that imaging offers huge potential as a platform for non-invasively measuring the pharmacokinetics and characterisation of small molecules, biologics and non-biological complex medicines. We think that by exploiting and using current capabilities, but also developing emerging opportunities and techniques, this will really facilitate faster translation into clinical development. Our overarching aims and objectives is we want to be discovery and research driven. We really want to challenge and optimise the use of imaging and testing and how we're doing it. We want to move from compounds to further transformational opportunities. We want to look at cell tracking. We want to really move on and try and understand and use imaging to the best of its capabilities. So before I answer a couple of questions, just really want to acknowledge everybody within the Medicines Discovery Catapult, everybody who's been involved in the work of these slides today, and also our collaborators, and especially to all of you for listening today. We thank you for that. Okay. So I think I have a few questions. So the first question is the measurements for the dye or the containing levels of drugs are very interesting, but doesn't the labeling of drug change its distribution and metabolism? So we've thought about this. It's a question that we're always asked for. So often an option is to do and look at the mass spec tissue imaging afterwards, but in our experience, then the dye and the zirconium label isn't interfering with that. In the studies that we do, we always have a vehicle treated group or an isotype control, for example. And so we were taking that into account with the control groups that we'd have. I'm just trying to look what else we have. For the blood-brain barrier spheroids, so it's how do you make your blood-brain barrier spheroids? Do you do 3D matrix? So we can probably come back to that offline. So that was a colleague of mine that has the expertise in this model. So we can go into that afterwards. I think that's all the questions for today. So there's another similar question about taking into account the imaging effects of the PK/PD. So as I say, we have controls there, either vehicles or isotype controls to look at that. So another question is, are labelling chemistries good enough? Where can we improve? So I think, so the chemistries that we use have shown some fantastic data and fantastic results. I think we can always improve. I think, you know, if we work with a networked approach, we share ideas, we share protocols, and I think certainly within the UK, this is very much an area where we think is very important for optimising the chemistry, for doing different methods, looking at different labels, etc.
Okay, thanks, Juliana. That's great. Thank you very much.
Perfect. Thank you.
So, thank you to all of our speakers today, to Al, Richard and Juliana, for steering us through the session on optimising compounds. I'm just going to share my screen. So next week, we're going to be focusing on chemistry and formulation, and we've got 3 talks for next time. So we've got Trevor Perrier from Domain X, who's going to take us through a case study of approaches turning fragment hits into drug candidates. We then have a talk by James Hitchen of Charnwood Molecular, and he's going to talk about synthetic chemistry aspects of drug discovery and early development. And finally, Alison Foster at Key Pharma about designing formulations for early stage preclinical and clinical studies. So thanks once again to the speakers today, and thank you all for listening, and see you again next week. Thank you.